Good afternoon, everyone. I would like to welcome you to this coffee science seminar. Next slide. My name is Professor Lo Hoffman. I'm a professor in meat science from the Center of Nutrition and Food Sciences as part of Queensland Alliance for Agriculture and Food Innovation from the University of Queensland. Right. I would like to start by acknowledging country. The University of Queensland acknowledges the traditional owners and the custodianship of the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australia and global society. I just want to start with some house rules. The seminar today is scheduled from 12 until 1 o'clock. After the presentation, we will hold a question and answer session. Throughout the webinar, please type your questions in the question and answer tab, not in the chat function. Thank you. And now it gives me pleasure to introduce Sean to you all. Sean is a PhD student at CNF within coffee, who has actually already received feedback and had his PhD and oral approved and is waiting for finalization of his PhD. So congratulations on that one, Sean. Sean did his previous master's study in food science and technology here at the University of Technology. And that's where he actually discovered his passion for edible insects. He worked on green ants. Sean's PhD project aimed to investigate the quality and safety of black soldier fly larvae reared on different food waste streams for livestock feed. In addition, he has also been involved in several other projects exploring the quality and functional property of insects intended for use as food and feed. Within his PhD, Sean has already published 6 peer-reviewed papers in Q1 journals and another 4 under review. And then he's also co-authored on some of the other projects and additional 4 papers. Sean, we look forward to your presentation. Thank you.
Thanks for the introduction, Love. I too would acknowledge the traditional owners and their custodianships of the lands on which we meet today and pay my respect to their ancestors and descendants. Thank you everyone for joining with us on the talk today. And for today's talk, I'm going to give you guys a bit of background on what's black soldier fly larvae, why we should care about it, and what my research has done to optimize and industrialize the black soldier fly larvae production, especially in terms of redirecting food waste to livestock feed. So before I get into the technical side of things, I want to share a bit of light on food waste and food security and the protein demand and problem. So annually, 7.6 million tons of food gets wasted in Australia. This is about 1/3 of the food that gets produced for human consumption. And globally, food waste costs around $990 billion on the global economy. And 92% of the food waste that gets generated directly goes to landfill, contributing to greenhouse gas emissions, thereby contributing to climate change. Approximately in Australia, 3% of the greenhouse gas emissions is occurring through food waste. Despite having all this food getting wasted, all this food that's been produced for human consumption, 1 in 9 people go to bed hungry every night despite having enough food on the planet. So now the protein demand and problem. So the protein demand for the current population of 7.5 billion people is around 202 million tons. And it's mainly contributed by cereals and meat. And this meat demand, especially for pork and poultry, is expected to grow up to 435 million tons by the year 2050. And the Australian protein market is valued to go around $19.9 billion by the year 2030. The problem with this protein demand is production of this protein. So currently, feed sources such as soybean oil, meal crops, and legumes are commonly used as feed sources for livestock rearing. However, this practice of livestock rearing is becoming challenging due to climate change and several other socio-environmental factors. So the need for viable and sustainable alternative feeds has been increasing. And this is where black soldier fly larvae comes into play. So black soldier fly larvae is commonly distributed in the tropical and subtropical climatic condition. And it is a saprophagus insect. That is, it can valorize on different organic matter, and it exhibits good feed conversion ratio. And it's been reported that, every single individual larvae can eat up to 5 times their weight. So this black soldier fly larvae goes around 6 stages with their life cycle. So the first 5 stages are the larval stages. They change in color, size, and weight. And the sixth stage is called the sixth insta larvae, or the pre-pupae. And that's when the larvae stop feeding on their substrate, try to move away to a fresh place. And later they hatch into pupae and become the adult flies. The best part about this larvae is that the adult flies, they're not a pest, and they are not a vector for other diseases. So they complete the life cycle, they want to reproduce, and then die, and the next life cycle begins. So what we do with this black soldier fly larvae is we try to bridge the gap. So we've got food waste in a hand, and we inoculate this food waste with black soldier fly larvae, which are 5 days old or 10 days old, pretty much in their first instars. And this black soldier fly larvae, they forage on the food waste, and they become rich in protein and several other nutrients, which makes them a good feedstock. And later, we eat the animals, and we again produce food waste, thereby creating a circular economy with this black soldier larvae. So in this slide here, kind of highlighted the key nutritional parameters for the larvae. So these larvae, they can vary in crude protein from 30 to 50%, and the fats around 15 to 49%, 15 to, yeah. And they have got a very balanced micronutrient profile, and also the amino acid is comprised with all the essential and non-essential amino acids. And they're considered to be a good source of lysine, and several other amino acids like tryptophan, threonine, and valine, they are found to be distributed in better proportions. And in terms of fatty acid, they're predominantly constituted by saturated fatty acids, with lauric acid being the major one, followed by palmitic and oleic acid. And the main important thing that should be noted is all these nutrient parameters is influenced by substrate, along with rearing conditions and other underlying factors. There's been several research done on exploring the nutritional properties of the larvae, and a lot of recent research is also available, which focused on feeding this larvae to different livestock, and then exploring the meat properties and whatnot. However, the commercialization of BSFL at feed source is limited, and the main reason for this limitation is because of the prevailing legislative framework. So currently, if you grow BSFL on vegetable substrate, it's allowed to be used as feed for swine, and then it's allowed to be used as feed for swine here in Australia. But if you grow them on organic waste streams, such as food waste, they are considered as restricted animal material and prohibited pig feed, and cannot be fed for ruminants and pigs. So this RAM and prohibited pig feed regulations exist due to lack of knowledge about the chemical and microbial safety of the larvae. So pretty much the regulatory bodies are concerned that, concerned about the risk of transmissible spongiform encephalitis when you grow these larvae on food waste streams and feed them to livestock. And there's been some research done around the microbial and chemical safety of the larvae. So in terms of bacterial hazards, most of the studies has focused on BSFL's ability to reduce bacterial pathogens like Salmonella and E. coli. And it was observed that, the growth rate and weight gain was not affected, and the feed conversion process was not influenced. But however, the effect of accumulation of these pathogens within the larvae was not explored. And in terms of mycotoxins, black soldier fly larvae's ability to reduce different mycotoxins into other metabolites has been explored. And heavy metals, they found that, heavy metals like cadmium and copper can accumulate, had higher bioaccumulation factors. But the one main thing, a couple of important things that needs to be observed from this previous research is that, all this research has been done on a lab scale. And all this research was mostly, focused on using formulated substrates. That is, you get your conventional feed ingredients, spike it up with contaminants, and then, you see what happens to the larvae. But however, that's not an actual representative of the food waste that you would, see in your trash can or see in a municipal bin. And from a commercial perspective, it is important to know what's actually present in the waste and what happens to the larvae when you feed those waste rather than, trying to formulate your own substrates. And also, if you want to translate the research outputs, then it's imperative that, we explore the safety of the larvae on a commercial scale setup with actual food waste streams that will be potentially used for rearing the larvae. So keeping that in mind, we have designed these experiments. So for my studies, we used 5 different waste streams. So one was the soy waste, and the other one was a customized bread and vegetable diet, which was kind of done to, have the idea of safe diet for the larvae. And then we had 3 other waste streams. So one was the supermarket waste, and the other was the waste obtained from childcare center. And then the third one was a mixture of food waste, that is food waste obtained from supermarkets, retail centers, and restaurants, fast food outlets, etc. So the trials that was done with soy waste and bread and vegetable diet was done at a rearing facility here in Brisbane. And from those trials, we collected 5th and 6th instar larvae. So 6th instar is the pre-pupae. And also we collected the 5th and 6th instar frass. While from the other 3 waste streams, we collected the 5th instar larvae and we subjected them to 3 different post-harvest treatments as well. So one was straight up homogenizing the live larvae, and the second one was blanching them at 100 degrees Celsius for 5 minutes. The third one was drying the blanched larvae at 80 degrees Celsius for 12 hours and then homogenizing them. And also we collected the frass from all these trials as well. So on all these samples, we had 4 main research objectives. One was examining the nutritional composition of the larvae, and the other was microbial safety, chemical safety, and also we wanted to, explore the possibility of using NIRS spectroscopy as a non-invasive rapid technique to, monitor different quality aspects associated with the food waste streams, larvae, and frass. So moving on to the first research objective, which was to, determine the microbial safety of the larvae. So here we examine the food waste streams, larvae, and the frass for the following list of pathogens that's listed on my screen. And we followed the Australian standards for that. So here I've got the microbial load of the food waste stream. So it was observed that, spore-forming bacteria, especially, Bacillus cereus and Clostridium perfringens, they were found to be relatively distributed at higher counts. And all these waste streams, they tested negative for Staph aureus, Listeria, Salmonella, and E. coli. And this slide here talks about the microbial quality of the larvae, especially the effect of 2 different larval instar, which is the 5th instar larvae that's commonly being used by the industry for commercial applications, and then the 6th instar larvae, which is the pre-pupae. So we can see from these results that the 6th instar larvae had reduced counts for spore-forming bacteria, and also we could see that, Bacillus cereus was reduced by 1 log units from soy waste streams, and in case of BV diets, it was 4 log unit reductions. In case of yeast and mold counts, the 6th instar larvae had a higher counts. This is probably due to the rearing conditions favoring the growth of the molds, and the organisms that were absent in the substrate were not present in the 5th and 6th instar larvae as well. So here I've got the effect of post-harvest treatments. So we can observe that blanching and drying treatments carried out here reduce the counts for Bacillus cereus below detection limits, and then with Clostridium perfringens, the counts were reduced as well. And the larvae again tested negative for Staph aureus, Listeria, Salmonella, and E. coli. So one important thing that we need to, the key findings from this chapter is that all positive organisms were within the maximum tolerable limits for Australian feed regulations, except for Clostridium perfringens. So we can see that, the spore-forming bacteria, especially something like Clostridium perfringens, whose spores are pretty resistant to thermal degradation, they seem to pose a threat to black soldier larvae's quality. So this here is the microbial load in the frass, because frass again in a commercial setup is being used as a fertilizer. So we'll have to make sure within the current regulatory framework, there isn't any specific regulation for frass in terms of microbial quality. But again, longer run, we still have to make sure that the frass is free of any pathogens that could potentially, affect the growth of the plants. So in terms of microbial load and frass, the fifth instar frass had counts that was pretty similar to the substrate. So normally you would expect that if an organism is present in your substrate and if the conditions are favorable for the growth of the organism, you would expect that, after 10 days of incubation, you would expect the counts to be higher, but the counts were not higher in this case. So we are probably speculating this is due to the antimicrobial action exhibited by the larvae. And obviously, the sixth instar frass had higher counts than the fifth instar because they undergo more bioconversions than the fifth instar larvae. And in terms of the frass from the other complex waste streams, they were all closely similar to the counts observed in the initial substrate. And again, the larvae, the organisms that were tested negative in the substrate and larvae were not present in the frass as well, which is an important takeaway from this slide. So moving on to the next research objective, which is the chemical safety of the larvae. And under this objective, we did two experiments. One was to examine the heavy metal distribution in the substrate, the larvae, and then the frass. And the other one was to see the presence of mycotoxins in the larvae when you grow them on different actual food waste streams. So here you can see the distribution of toxic heavy metals in the different larval instars. So we can say that the sixth instar larvae had higher concentrations of copper, magnesium, and zinc. And it's been reported that, some of these heavy metals aid the larvae with certain specific physiological functions. However, the more important message is that the toxic heavy metals were not distributed at higher concentrations in the sixth instar larvae. And overall, in both the fifth and sixth instar larvae, the concentration of these heavy metals were below the maximum tolerable limits established for animal feed, both under EU and Australian regulations. The reason why I state EU regulations is because they've got slightly I'd probably use the word edge in terms of sorting out the regulatory framework and commercialization scale for insects as food and feed. And the limits are within both the EU and Australian federation regulations. So in terms of distribution of heavy metals in BSFL, especially examining the effect of post-harvest treatments, so blanching that we carried out was observed to cause leaching of manganese, zinc, cadmium, and chromium. But it should be noted that these leaching effects were not significant, and there was found to be binding of these heavy metals with chitin and other protein structure, which has caused an increase in concentration of copper, arsenic, cobalt, and nickel and lead in blanched samples. So in case of dried samples, significant increases in concentrations were observed for zinc, manganese, and copper. So this is probably because of the way the samples were blanched and then dried. And again, the key important takeaway from the slide is that despite the higher concentrations observed for the dried samples, the concentration of toxic heavy metals that is regulated was below the maximum tolerable limits, which again says that the larvae is safe for heavy metals. And moving on to the next one, the larvae was safe for the mycotoxins as well. They were all below the limit of detection. And in the table here, I've got the concentration of heavy metals in the frass. So obviously, sixth instar frass had higher concentration than the fifth instar frass. This is because of the longer bioconversion process. And there are certain standards for fertilizers stating that the concentration of heavy metals, especially arsenic, lead, and cadmium, cannot exceed certain limits. And again, the frass that we obtained from our trials did not exceed these limits, saying that they are safe to be used as fertilizer under Australian regulations. So moving on to the next research objective, which was to explore the nutritional composition of the larvae reared on different food waste streams. The whole rationale behind this chapter is that we have done post-harvest treatments to make sure the larvae is safe against microbial contamination. But we also wanted to make sure that those processing treatments do not compromise the nutritional quality. So here, to start off with the effect of instar, we observed that sixth instar BSFL had higher crude protein. This is probably because of the chitin, and then the fat content in the sixth instar larvae was found to be reduced. This is because the larvae, when they grow old, they stop feeding and move away from the feed, as I said earlier, and they rely on their fat sources to support their life, and that's why you see a reduction in fat content in the higher instars. And the protein content, if you could calculate the chitin specifically present in the larvae, and then just look at the actual protein content, you would see that you would expect that there shouldn't be any change in the protein content after a certain stage. And in terms of discussing about the effect of processing, it did not significantly alter the nutritional qualities for most of the parameters that was analyzed in our studies. And the blanching that we carried out based on the IPIF, which is International Platform of Insects as Food and Feed. So we did the blanching based on the guidelines, and that did not affect the mineral content. So moving on to the distribution of amino acids. So we found that distribution of certain amino acids was found to be influenced by the larval instar, with feed playing a substantial effect. So the effect of instar on amino acids could be attributed by the role played by the specific amino acids at different life stages of BSFL. And drying was found to influence amino acid concentration for certain amino acids. Because you could see that with in the relative distribution chart that I've shown here on the slide, you could observe that for certain amino acids, drying did not have any effect when compared to their blanched and controlled samples. But for certain amino acids, they either resulted in a decreased content or decreased concentration, or resulted in higher concentration. Overall, the blanching and drying experiments carried out as part of a study did still result in a larvae with a nutritional with an amino acid composition that is comparable to that of other feed ingredients such as soybean meal and oil crops. So the fatty acid profile, as I was mentioning earlier, it was predominantly constituted by saturated fatty acids, followed by polyunsaturated and monounsaturated fatty acid. So the polyunsaturated fatty acid distribution was varied with different larvae. Larvae grown on different substrates, because the larvae doesn't have the enzyme precursor needed for de novo synthesis of unsaturated fatty acid. However, the larvae uses the carbohydrate content in the substrate for the synthesis of saturated fatty acid, and that's why you see that the larvae that was grown on the bread and vegetable diet has a very high saturated fatty acid contribution, especially for lauric acid. And the fatty acid contents in terms of, the effect of drying in terms of fatty acid content was slightly found to be negatively affected. But again, it is still comparable to that of conventional feed ingredients. So this year, what I've done here is we wanted to identify the most important nutritional parameters that affects the overall quality of BSFL. So people who have been involved in BSFL research would know that most of the nutritional parameters is heavily influenced by the substrate that's used for growing them. So what we did here was we employed a chemometric technique, which is a multivariate statistical technique, and we tried to use different nutritional parameters that we measured in our studies, which includes all the proximate analysis, and then different amino acid composition and fatty acid composition all together as variables. And we tried to we use them as variable to relate them to the different feed sources used to rear these larvae, and we obtained the PLSTA models to classify the larvae based on their feed sources. And with the plot that I've shown here, you could see that the larvae formed into distinct clusters based on their feed sources, and also you could see that there are subclusters formed between certain groups, and this is because that's because of the effect of blanching and drying that was done to the samples. And from these PLSC analysis, we looked at the loadings plot, and we found that separation was governed by fat content and fatty acids including decanic acid, lauric acid, myrcic acid, and minerals such as magnesium, calcium, phosphorus, potassium, and sulfur. So what it essentially means that these were found to be the most important variables within the feed source that influences the that particularly influences the nutritional composition of the larvae. And in terms of protein content and different sets of amino acids, there were certain set of amino acids which was found to negatively correlate with the separation, stating that there are certain amino acids that is not regulated by the feed source, and the larvae kind of have genes to regulate their own synthesis of those specific amino acids. So moving on to the final research objective, which is the use of near-infrared spectroscopy for nutritional and safety profiling of BSFL. So I'm going to try and give a one-liner about near-infrared spectroscopy. So it's a molecular, it's a vibrational spectroscopy technique where you measure the vibrations of different molecules, and these molecular vibrations will change with sample presentation when there is compositional changes and things like that. So you try and map those vibrational changes with your reference data set, and you try and predict them. So for this research objective, the first experiment we did was a traceability study where we tried to, see if we could apply this technology for black soil larvae. So for the traceability study, we tried to, trace the larvae based on their larval instars, based on their larval physiology, and also to see if we could, use NIR spectroscopy to trace the larvae based on the feed source used to grow them. So in the first scores plot, which is on the left side of the screen, we could see that the larvae form two distinct clusters. The one that's in the red is the 6th instar larvae, and the one in the blue is the 5th instar larvae. So we could see that, the NIR instrument could differentiate the larvae based on their physiological age. And after that, we classified the larvae based on the feed sources, and we could see that, the larvae could, we classified the 5th and 6th instar separately based on their feed sources, and we could see that, the larvae were grouped into two different classes based on their feed source. And the main takeaway from this traceability study is that the physiology of the larvae plays a much important role than the feed source used to rear them. So here, so after confirming that, we could use NIR for traceability, NIR for BSFL applications, then we moved on to, predict the nutritional quality and microbial safety of the larvae. So we tried to, predict the nutritional quality such as sugar, starch, crude fat content, and several other parameters for the larvae and the frass. And we got good cross-validation statistics for measuring ADF, which suggests that we could have quality control applications for measuring ADF fiber content. And we obtained moderate cross-validation statistics for determining other nutritional parameters such as total carbon, crude protein, crude fat, starch, sugar, and NDF fiber, which suggests that you could use them as a screening tool. You could use NIR as a screening tool for these particular parameters. And for microbial safety, we obtained good cross-validation statistics for the models developed for predicting Eastern moles in feed and Bacillus cereus in frass samples, and Clostridium perfringens in frass samples, which suggests that these models could be used for predicting these specific organisms in specific samples for quality control applications. And we obtained moderate cross-validation statistics for the other models that we developed as part of the study, which again says that you could screen if a sample is particularly good or bad at a particular concentration. So the main advantage of using NIR for the screening purposes is that they are non-invasive and they're very rapid. You don't have to spend a lot of hours in the lab trying to do all the analysis. It's just on the go. You scan the samples and you have a reference data set, and you try and predict the nutritional quality and microbial safety. So to give off brief general conclusions, the larvae reared on different food waste streams were found to be safe for most foodborne pathogenic bacteria. However, there is risk of spore-forming bacteria in the larvae. And 6th instar was found to be lower in pathogen loads than the 5th instar. The blanching and drying treatment, however, was found to reduce the counts for the 5th instar larvae as well. So the larvae and frass was found to be safe for all chemical contaminants examined in the study. In terms of nutritional quality, slight changes were observed for different instars for crude fat and fatty acid contents. The minerals and amino acids were partially influenced by the instar and the feed. The drying conditions followed negatively affected fatty acids, concentrated certain minerals, and in terms of the amino acids, the effects were varied. But again, the most important thing is that, despite these changes, the nutritional quality of the larvae is still comparable to that of commercial feed ingredients. And the NIR's technology can be used for rapidly monitoring different quality aspects of BSFL. So this could be used for both quality control and screening-based applications in commercial production lines. You could install these instruments within the production line, and then they'll be there to rapidly monitor the quality. So in terms of project impact, we were able to effectively monitor and improve different aspects of product quality. And the findings of this research could help improve process efficiency at different stages of the production line. But yeah, you could, based on the substrates you're dealing with, you will know what specific contaminants or risks you need to focus so that your product is in line within the regulatory framework. And also the rapid non-invasive, with the NIRS, we found that, we could have a rapid non-invasive inline monitoring process, inline monitoring of process and product quality. This can assist in reduction of production cost and make sure the product is always at the targeted quality. And with this research, we could identify hazards and critical parameters for controlling BSFL quality. This will enable us to ensure compliance with current regulatory framework and other certifications that are needed to commercialize the larvae. And overall, we also, this has also helped us understand the impact of processing on insect product quality. And understanding this will help us create guidelines for good manufacturing practices for commercial BSFL facilities. I'd like to acknowledge my advisors, and I'd like to thank the technical staff who's been guiding with me for the microbial experiments, and I'd like to thank all the staff at Gotera and Flyfarm for helping me with the experiment sample collection and everything. And I'd like to thank Coffee AGFS within the University of Queensland, and I'd like to acknowledge N Food Waste Cooperative Research Center for funding this research project. Yeah, thanks for listening to the talk. I'm gonna hand it over now to Love to take us to the question and answer session.
Thank you very much, Sean. I'd just like to remind everyone to please use the question and answer if you have any questions. I can see from the attendance list, and I'd like to welcome some of the insect producers from Australia. I see there's a couple of different producers online, as well as other visitors from outside the UQ scenario. The questions coming, some I'll answer, but some are specific to Sean's study. Sean, and I want to ask you to answer the first one, and that is, in your work, did the supermarket waste feed substrate contain any meat products?
Yes, yeah, it actually did contain meat products. So supermarket waste, as such, had meat produced, dairy products, and everything else, that's kind of not really shelf stable was there in the supermarket waste.
The other one is a question around the influence of the length of blanching time on amino acid and fatty acid compositions, and is there a way that you could manipulate the blanch? If there is there a way to manipulate the blanching time to reduce or minimize impact?
So in our studies, the blanching time we carried out was just been, like an I wouldn't use the word trademark, but it was something that was suggested by the IPIF. But I did come across certain other studies where they tried out different blanching times and drying treatments, and in their studies, they didn't observe any change in the amino acid composition. So I wouldn't expect that you would be able to, stimulate the amino acid composition of the larvae just by manipulating the blanching and drying conditions. Because there has been some very recent research which found out that, there are genes within black sulfate larvae for regulating certain amino acid production, and they do have a mechanism for upregulating and downregulating certain specific amino acids based on their requirements. And these requirements will vary depending upon the type of substrate they are grown on and then the processing conditions.
Thanks. I'd also like to just expand on that one, and that is, we do know that the type of waste being fed to the black soldier fly influences their fatty acid profile, similar to any monogastric animal. So if I was to feed them on fish waste, I would have quite a high level of polyunsaturated fatty acids. And one of the problems you then have is if you blanch it for too long at this high temperature, you actually end up with destroying some of the fatty acids or getting a oily layer on top of the water. So that is something to look at further that we would be looking at. The other one question that is around there is, could you expand a bit about the shelf life of this insect meal? And is it possible to increase the shelf life for the products?
So as such, I mean, yeah, in our studies, we didn't look at the shelf life, shelf life of these products, but I would imagine that, I mean, I have come across certain studies, where they have done some preliminary shelf life studies for the dried larvae, and they have been pretty much focusing on just the total microbial count and whatnot. So the dried larvae, they seems to be shelf stable for close to like the studies. I'm just giving this number from the top of my brain. I remember, they were found to be shelf stable for close to 60 days at room temperature, the dried larvae. I can't remember the drying conditions, but yeah, that's again a domain that needs to be further researched.
Right, and then there's a question around, is there an interest in carrying out the further studies on feed substrates like pig manure? And I'd like to answer that one, and that is that, like any animal production system, as good as the feeders will influence your productivity. And within the whole black soldier fly scenario worldwide, there are two different thought processes on this one. And the one is to maximize the production of the black soldier fly larvae. And to get that, the better the feed that you feed, the better the growth and the amount of black soldier fly you'll get. The other one is to utilize this uniqueness of the black soldier fly larvae that can upcycle wastes into frass or a compost more readily, and also still have an additional product, which would be your black soldier fly larvae. So there has been quite a bit of work done utilizing others, such as pig manure or chicken manure or even ruminant manure. They do it, but obviously because these fecal samples, and that also includes human fecal samples, have had a lot of the nutrients already absorbed by the animal or person consuming them. So the nutritional profile of that waste isn't so good, but it will still, so you won't have as fast growth rates or production. So I suppose at the end of the day, it depends on what is your end target. Is it to maximize production of black soldier fly larvae, or to utilize an alternative green method, a biological waste, to utilize it in a better manner? I see whilst I've been talking, some more questions have come in. And Sean, going back to your actual study, the question is, what could be the source of these heavy metals? Could they be from the food waste substrates? Because after all, these food wastes all meet the minimum requirements to be sold as human food.
Yeah, so around that, like Lau said, this food waste was initially food that was considered to be safe for human consumption, and you would expect that the heavy metals were, the concentration of these heavy metals would be low. But what I'm guessing is the food waste, after it's been collected from the facility, they are all put together, and then they undergo some sort of natural composting, which can result in certain levels of leaching of these heavy metals. And I'm speculating that this could have probably resulted in increased concentration of these heavy metals. But again, saying that, in for both the frass samples and then the larvae samples, the concentration of these heavy metals, even though was varied, it was still within the maximum tolerable limits for respective fertilizer guidelines and then the animal feed guidelines. So I guess that's the more important part to look at, from a regulatory safety perspective.
I see another question that's come in is, how far can this research be stretched into producing full meal animal feed, such as pellets?
Yeah, so there have been a lot of, even within Australia, there are a lot of industries, which are manufacturing black soldier larvae pellets. And as I was talking earlier in my presentation, currently you can use these feed pellets for aquaculture, and then for poultry and for your pets. So that's where the market is currently at the moment. But we realize the potential this has, especially if you want to use for non-ruminants around, for pig feed, and that's what we want to achieve with this project. We with the research outputs we've got, we found that the larvae seems to be safe within most of the existing regulations for animal feed, and then we want the product to not just stay within aquaculture and pet feed. We want it to expand for swine feed and also eventually into ruminants.
I think if I could also come in there, is we're not proposing feeding black soldier fly as a single component to animals.
Yeah.
We're seeing it as an alternative feedstuff to put into a balanced trial. And on that one, we're at the moment within this project, we're actually looking at using black soldier fly as a partial replacement for soya in poultry diets, as in broilers, as well as in egg layer diets. And it's at the moment, most of the black soldier fly larvae produced is going into the pet food industry because of the value of the pet food industry. If you were to work out a bag of dog food per ton, the cost, it is something like 10 to 20 times higher than the same balanced diet that you feed to a pig. So that's where we're going. One of the questions that came up is for you, Sean, what would you say were the limitations of your study, and what would you propose the direction for future research?
So in terms of limitations, one thing that I could say is, especially around the NIR aspect of it, we've shown proof of concept, but for us to have an fully commercial, if you want to implement that, we'll need to diversify the sample set and things like that, and that would be a limitation, and also the future direction for research.
Okay, so within this scenario, if I can expand on that, we presently have PhD students looking at the use of black soldier fly larvae in various animal diets as part of a balanced diet. We also have a student looking at an aspect that is actually gaining a lot of attention worldwide, and that is microplastics. In this food waste, nearly all the food waste comes in some form of a plastic wrapping. So we're looking at that as well. And then the other is a whole new direction that we're going into is, can we add more value to the black soldier fly per se? So we're looking at various technologies to separate the chitin from, as well as the fatty acids and the amino acids. And the initial work has shown that if we do that, if we remove the chitin and the fatty acids, the amino acid profile that we have left is so well balanced that it will most probably have a market in the human elite athletes, sportsmen scenario as a nutritional substrate. So there's a lot of different movements going in this space. One of the others is where they're looking at alternative wastes and sometimes doing a pre-treatment to the waste before feeding it to the black soldier flies. As an example would be doing some form of fermentation and then feeding the larvae on the resulting product. I see there's also been additional questions that are coming up. And that is, would there be any issues with risk of mad cow disease prior transmission from feeding meat animal waste to the larvae and then reintroducing it into animal feed? In other words, is in your mind, Sean, is this a real life problem or not?
I mean, there has been this larvae, how it has been responding to any contaminants for that matter is it's got a microbiome within it, and it's got a way to metabolize or either excrete these toxins out of them. And I have come across one particular research where they tried to use this larvae to kind of reduce an organic waste that had viral particles in it. And the larvae, it still came out clean and didn't have any viral particles. And the regulations that are currently in place, the RAM regulations and then the PPF regulations, it is there because of this risk. And we did find out from our research that it is safe for most of the other common risks that's been highlighted from under the animal feed perspective. And about the issue of prion, I would imagine that the larvae would treat this like any other contaminant that it's been dealing with, because in terms of mycotoxins and heavy metals, even though it enters the system, it somehow excretes it. And I'm guessing if at all the prions are present in the substrate that's been used, they are going to they're going to excrete it or not accumulate in the first place at all.
Yeah. So, Sean, another question that came up is your study was actually focused on industrial production of black-solid fly. But there is also another group of people that actually have black-solid flies, and that's on a household level, where they use black-solid fly within their recycling bins or compost bins. And because it occurs all over Australia. So the question is, how would you increase the production of black-solid fly larvae, and what should we do to optimize their production performance during winter, especially for the household rearing and usage?
So that I mean, if we want to grow this larvae under the household rearing setup, winters can be actually a bit challenging, because as I was saying earlier, it's just not I've cracked this particular formula and this is gonna give me a very high quality larvae. It's all these factors works together in a synchronization. So we'll have to find you got to have a balance of everything, and maintaining temperature is one key factor that needs to be done to ensure the survival and growth of the larvae. So from an household perspective, if it's going to be challenging, I mean, you could get heaters and other humidifiers and incubators to increase the temperature. But again, from a household perspective, it just increases your production and operation cost. And I would again go back to what I was saying earlier. It's just that all these factors work together in synchronization, and we'll have to do something to make sure we keep them happy in terms of the temperature aspect.
Yeah, of course. The temperature is an important one, and that actually gives the Australian industry an advantage compared to the European industry, because we have warmer temperatures this side. In Europe, they have big challenges. They have to heat the facilities during winter. And that brings up the whole question when you start using black-solid flies as a green methodology to recycle waste, but then you use not necessarily green energy to heat up the facility or to produce it. Similarly, when you start using quite aggressive solvents to extract the various chitin, protein, and that, then it becomes quite a challenge, because you're using that green image. So another question that came in is, we still have a few minutes. Do you think that black-solid fly larvae promote a microbiome within their substrate rather than having an internal microbiome?
Yeah, so there's been a whole lot of research done around that space. So the larvae, they do have a core gut microbiome, and it's been found that the larvae rather stimulates the microbiome of the host substrate rather than letting the substrate's microbiome influence it. Especially, there was one particular research which focused on the fungal communities, and it was observed that the larvae's microbiome and then the internal fungal communities kind of influence the growth of the presence of other fungal communities in the substrate. So it's the other way around. The larvae's core gut microbiome plays a significant role in influencing a lot of metabolic reactions and then influencing the host substrate's microbiome.
Okay, thanks for that one. What's fascinating is the fact that actually these black-solid fly larvae grow in putrid, a lot of times putrid substrates, yet they survive. And there's also an interest within that. Can we actually use these antimicrobial, antifungal properties when we place them in poultry diets, for example? And some of the research I was involved in, we actually found that we could partially replace, if not fully replace, some of the antibiotics in poultry diets with black-solid flies. And a lot of that is, of course, linked to the C14 fatty acids itself. Sean, I think it's time to wrap up if there are no more questions. I'd just like to go to the next slide, please, Sean. I'd just like to invite everyone that you could use the code on the screen at the moment, scan it, and from there you'll see all the Coffee Science Seminars webpage, which you are welcome to peruse at your own convenience. Similarly, if you would give us a few days, this presentation will also be on the Science Seminars. It just remains for me to once more thank Sean for his presentation today, as well as all of you who are attending this presentation. Have a good afternoon and stay safe. Thank you. Bye.
Thanks everyone.